Good morning, everyone. Our lesson for today is light independent stage of photosynthesis. After the energy from the sun is converted into chemical energy and temporarily stored in ATP and non-pH molecules, the cell has the fuel needed to build carbohydrate molecules for long-term energy storage. The products of the light-dependent reactions, ATP and non-pH, have lifespans in the range of millions of seconds, whereas the products of the light-independent reactions, carbohydrates and other forms of reduced carbon, can survive for hundreds of millions of years. The carbohydrate molecules made will have a backbone of carbon atoms. Where does the carbon come from? It comes from carbon dioxide, the gas that is a waste product of respiration in microbes, fungi, and animals. All plants having capability of photosynthesis are universal providers because they capture energy from the sunlight and prepare their own food by the process of photosynthesis. They provide the animals and human beings with the food to eat. In plants, carbon dioxide enters the leaves through stomata, where it diffuses over short distances through intercellular spaces until it reaches the mesophyll cells. Once in the mesophyll cells, carbon dioxide diffuses into the stroma of the chloroplast, the site of light-independent reactions of photosynthesis. These reactions actually have several names associated with them. Another term, the Calvin cycle, is named for the man who discovered it, and because these reactions function as a cycle. Others call it the Calvin-Benson cycle, to include the name of another scientist involved in its discovery. The most outdated name is dark reactions, because light is not directly required. However, the term dark reaction can be misleading because it implies incorrectly that the reaction only occurs at night or is independent of light, which is why most scientists and instructors no longer use it. The products of the light reactions, which is ATP and non-pH, shall be used in the Calvin cycle or light-independent reactions. The light-independent reactions also occurs in the stroma. This is a quick overview of what occurs during photosynthesis. The light reaction is the initial stage of photosynthesis, which traps light energy to produce ATP and non-pH, whereas light-independent reaction is the second step of photosynthesis, which utilizes the energy from ATP and non-pH to produce glucose. The light-independent stage is composed of three stages or phases. Phase number 1 is the fixation of carbon dioxide or carbon dioxide fixation. Phase number 2 is the reduction of 3-phosphoglycerate to G3P. Phase number 3 is the regeneration of RUBP from G3P. In the stroma, in addition to carbon dioxide, two other components are present to initiate the light-independent reactions. An enzyme called ribulose bisphosphate carboxylase or Rubisco, and the three molecules of ribulose bisphosphate or RUBP. RUBP has 5 atoms of carbon flanked by 2 phosphates. Rubisco or ribulose bisphosphate carboxylase catalyzes a reaction between carbon dioxide and RUBP. For each carbon dioxide molecule that reacts with one RUBP, 2 molecules of another compound, 3-PGA or phosphoglyceric acid, is formed. PGA or phosphoglyceric acid has 3 carbons and 1 phosphate. Each turn of the cycle involves only 1 RUBP and 1 carbon dioxide and forms 2 molecules of 3-PGA. The number of carbon atoms remains the same as the atoms move to form new bonds during the reactions. This process is called carbon fixation because carbon dioxide is fixed from an inorganic form into organic molecules. The second stage, ATP and NADPH are used to convert 6 molecules of 3-PGA into 6 molecules of a chemical called glyceraldehyde 3-phosphate. That is a reduction reaction because it involves the gain of electrons by 3-PGA. Recall that a reduction is the gain of an electron by an atom or molecule. 6 molecules of both ATP and NADPH are used. For ATP, energy is released with the loss of the terminal phosphate atom, converting it into ADP. For NADPH, both energy and the hydrogen atom are lost, converting it into NAD+. Both of these molecules return to the nearby light-dependent reactions to be reused and re-energized. For the third stage, regeneration of RUBP. Interestingly, at this point, only one of the G3P molecules leaves the Calvin cycle and is sent to the cytoplasm to contribute to the formation of other compounds needed by the plant. Because the G3P exported from the chloroplast has 3 carbon atoms, it takes 3 turns of the Calvin cycle to fix enough net carbon to export 1 G3P. But each turn makes 2 G3Ps, thus 3 turns make 6 G3Ps. 1 is exported while the remaining 5 G3P molecules remain in the cycle and are used to regenerate RUBP, which enables the system to prepare for more carbon dioxide to be fixed. 3 more molecules of ATP are used in these regeneration reactions. So to recap, in stage 1, the enzyme Rubisco incorporates carbon dioxide into an organic molecule, 3-PGA. In stage 2, the organic molecule is reduced using electrons supplied by NADPH. In stage 3, RUBP, the molecule that starts the cycle, is regenerated so that the cycle can continue. Only 1 carbon dioxide molecule is incorporated at a time, so the cycle must be completed 3 times to produce a single 3-carbon Ga3P molecule and 6 times to produce a 6-carbon glucose molecule. Thank you very much.